what are genes and how they are transferable, what are acquired traits, what are recessive traits, we have discussed. Okay, today we're going to discuss that why do Mendel choose pea plant for his experiment. Okay, why do Mendel choose pea plant for his experiment? The first question start that we are going to study the monohybrid cross and dihybrid cross and their conclusions that were very important. Okay, so we're going to discuss that why do Mendel choose, first of all, Mendel choose pea plant for his experiment. Pea plant, choose pea plant for his experiment, Okay, the reason behind is that pea plant is actually bisexual. Okay, and this will help us, or this will allow us to we can say simply here that this will allow us to do the self pollination as well as cross pollination. Okay, there are so many points that why do Mendel choose pea plant for his experiment. The first one is that pea plants has large number of traits. And what is the benefit of large number of traits? It has large number of traits. So what's the benefit of large number of traits? That you can see that the plate,
Differentiate.
yeah, we can differentiate and we can study them. What we are studying, actually, we are studying traits only, that if I'm just crossing a tall plant with a small plants, how many generations or how many plants are going to be tall, how many plants are going to be dwarf. Okay, so if I talk about that, pea plants are bearing large number of traits, means several traits are there: dwarfness, short plant, okay, dwarf plant, okay, or tall plant, or we can say round seed, wrinkle seed, the color of seeds, there can be different colors of seeds, flowers. Okay, so large number of trait is going to help us to study. Okay, actually we are just studying the trait, so if there are more and more traits, so it will help us to study more betterly. Okay, got it. The second reason behind this is that pea plants have very short lifespan, means it is not going to have large means life of pea plant is very short, or it can grow in 2 to 3 months. Okay, or it can bear seeds in 4 to 5 months. So it is very helpful for us that if we are using pea plants, so several due to short lifespan. Don't write it, I will help you to write it. Okay, just I'm going to explain you right now. So due to short lifespan, we can say that we can study several generations. So several generation can be studied. Okay, several generation can be studied. Okay, can be studied. So what actually we are doing, that if we took a plant, okay, that is, or that is going to have large number of or we can simply say that if there is a plant, okay, to have very large, okay, I'm just having, okay. Second one is that it is going to have short lifespan, and because of that short lifespan, several generation can be studied. Third one is that it bears bisexual flowers. And what is the benefit of bisexual flowers? That this bisexual flower actually, okay, because of this bisexual flower, we can say that it can easily self-pollinate. Okay, it can be self-pollinated. So if we want to do the self-pollination, the self-pollination is easily possible. Okay, got it. So bisexual flower help in self-pollination. Self-pollination, pollination. Okay, got it. And if it is going to have, okay, self-pollination. And if I'm talking about that, if if we want to do the cross pollination, then it bears large stems. So if a plant is going to have very small pollen grains, so we cannot remove it. Okay, this is very difficult to remove them. But if it is bearing large stamen, so they can be easily cut and removed. And if if we are able to easily cut or remove the stamen, which is a male reproductive part of the flower, so what is going to happen here is that we are going to have only pistil, the female reproductive organ. And from, if we want to do the cross pollination, we can transfer the pollen grain from other plant or other flower. Okay, and we can transfer the pollen grain here, and it can further germinate here. So if we want to do the cross pollination, so cross pollination is possible due to large stamens. So large stamen can be removed, okay, easily, and which will help us. And what is going to help? It will help us to do the cross pollination, okay, which will help us in doing cross pollination. You understood? Understood? So
Yes, sir.
it has large number of traits. It be a large number of traits. Okay. Short lifespan. Due to short lifespan, several generation can be studied. It is bisexual, so which help us to do the cross self pollination. And it bears large stamen, which can be easily removed and cut. And we can simply say that this is help us in doing the cross pollination. Okay, got it. So cross pollination, self pollination is also possible. The third one is that it bears large number of it bears it bears large number of seeds. And large number of seeds, what is it going to do? If I'm saying that it bears large number of seeds, so large number of seeds help us to get the authentic conclusions. Okay, if I want that how many plants, for example, if I have 400 seeds. Okay, so if a plant is going to give you 400 seeds, so I can calculate the authentic conclusion that how many out of this number of seeds, we have this number of wrinkled, this number of round, this one of wrinkled green, this one is round yellow. So we can, large number of seed is going to help us to get the authentic conclusions. Okay, got it? We can get the authentic conclusions since it bear large number of seeds. If there is only two types of seeds it is bearing, just suppose, imagine. So how can I say? So we can, if we want to decide that which or how the seeds are being beared, okay, how the traits are being transferred. So we need to do the large number of experiments, okay. And since it bears large number of seeds, so we can easily get the authentic conclusion. You understood? 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 Tell me fast.
Yes.
Yes, sir.
Okay. So it bears large number of seeds. Okay. Now moving to the next part is, just a minute. So please note it down that why do Mendel choose pea plant for his experiment? So I'm just going to give you here, just a minute. So please note it down. Why do Mendel choose pea plant for his experiment? The answer is Mendel chose pea plant for his experiment because availability of detectable contrasting characters, several characters like height, shapes of seeds can be studied. Okay, so this is the availability of characters are there. Okay, short lifespan. Therefore, several generation can be studied in a short period of time. This is very important. This is the most important, short lifespan. Okay, availability of contrasting character. Okay, these 2 points are very important. This is the main reason why he chose. Other plants also have, okay. Uh, there is cross pollination, means if the flower is bisexual, normally allows self pollination, but cross pollination can be carried out as it bears large stamen, which can be easily cut and removed. So please note it down fast. And when you're done, please let me know. Done?
No,
Jovita,
sir.
Note it fast.
Sir, writing.
Jaslyn, are you writing?
Yes.
Note it fast. I will check your notes after the class, okay? And if you are writing or not, let me check whether if you are doing okay or not.
Sir, done, sir.
Done, sure.
Yes, sure.
Okay. So this is the reason why do Mendel choose pea plant for his experiment, okay? Pea plant was a good plant for him. And how many years he was doing this experiment? For 8 years. How many years? He do this experiment, continuous experiment for 8 years approximately, okay? To know the
Sir,
how the
how
traits.
old was Mendel when he was doing this experiment? Started.
Actually, I don't know, but he will be in ठीक-ठाक age.
Like in his 20s or like as a child or some like it.
Yeah, yeah. No, no, it's not 20. He was aged during that time. Okay, so
Hmm.
done.
Yes, sir.
Yes, you, Jobita, John, Amar, Madiha, done.
Sir, writing.
Noted fast. The next is Mendel did monohybrid and dihybrid cross. Okay, and what is monohybrid cross and what is dihybrid cross? We're going to discuss this one. This was Jaslyn and you everybody. This has been written in the notes. Okay, so if you are taking much time in writing, so you can complete it from there. Okay, should I move forward?
Yes, sir.
Okay, done. Almost done.
Yes, sir.
Madiha done. Madiha.
Done, sir.
Okay, so let's move further. So what I'm talking about here is that mono hybrid cross. We are just going to study mono hybrid cross. Okay. Actually, what is mono? Mendel did experiments, okay, by crossbreeding the two different plants, okay, and he find different conclusions and he find and give some laws on the basis of that one. And what were those laws we're going to discuss? First of all, what is mono hybrid cross? So mono means single. Mono means single, and hybrid means transformations of character. Okay, hybrid, you know that if you are doing something by your own by transferring the something from one plant to another, that is called hybrid. So monohybrid means the cross in which single character means, like if I'm talking, there can be so many characters. Okay, like height is a character, color is a character. Okay, so he in monohybrid cross he only discuss the height. Okay, and height has at least two traits. Every character has at least two traits. It can have many traits, but it has at least two traits. Okay, one is, here in case of height we have two traits. One is tallness, another one is dwarfness. Got it? So if I'm talking about that monohybrid cross, so in monohybrid cross they are the two traits. Okay, one is tallness, another is dwarfness. So this is called this is called what we called it, traits, pair of traits. So every character has at least one pair of traits. So what is monohybrid cross? Monohybrid cross, if I'm talking about monohybrid cross, so what is monohybrid cross? The cross in which two uh, two traits, or we can say that the cross in which one one character character or one pair of trait, one pair. This is a pair of traits. Okay, in one characters we have, like height is a character. Okay, and one pair of traits is studied. Okay, is studied. We called it monohybrid cross. Understood? Understood?
Yes.
Yes, sir.
Yes.
Now, what he did actually, he took a homozygous tall plant. Homozygous tall plant. Homozygous means whose mother and father, who are male or female, plant is tall. Okay, though both are tall. And he took a homozygous dwarf whose mother and father both are dwarf. Okay, or whose male or female parent are both are dwarf. So this is approximately 6 feet plant. This is 6 feet, and this is approximately 1 feet plant. Okay, and he did the cross pollination. He did the cross pollination. Okay, keep in mind always that in the first generation which is obtained, he did the cross pollination. Cross pollination. Okay, cross pollination was done. Okay, and when he did the cross pollination, then what happened? When he did the cross pollination, then what happened? Actually, he got all the same plants of 6 feet. Okay, they were all same plants of 6 feet, and none was intermediate. Okay, they were all tall plants. All plants were tall. If I'm doing 100 plants, so 100 plants were tall. Okay, I'm just drawing 4 here, but there is not specific. You can see. Okay, got it. So all plants were tall. And why were tall? There was no intermediate, and this generation is called F1 generation. F1 generation, or we called it first filial generation. Okay, so always keep in mind that whether it is a monohybrid cross or whether it is a dihybrid cross, if we are getting the same type of characters, same type of things, same type of gene we are getting, okay, like I'm talking about capital T, small t, capital T, small t, means all are same. They're not different. Means all plants are of same characters. If it is round yellow, then all are round yellow. Okay. So that generation is considered to be as F1 generation, because in F1 generation, what is a monohybrid cross or dihybrid cross, we get the same kinds of plants. All were same. Got it? Got it? And we obtain this by doing cross pollination. So F1 generation is obtained by F1 generation is obtained by cross pollination. Keep in mind always. Okay. So in F1 generation, we did the cross pollination. Okay. Got it? Got it?
Yes, sir.
Sure. Okay.
Yes.
Now what we are going to do here is, have you made this one, everybody in the class? Have you made this one?
Mm.
Draw this one. Draw this one first. Draw this one. And if what you can think here, what you think here, that what are the possible things that he could think here, that all plants were tall, there was no intermediate. So what is that? What he can conclude from this one? There is no intermediate. So actually he can think that the gene for which a plant, or the factor, Mendel called factor, which we in modern term call gene. Okay, so Mendel's factor in modern term is called gene. Okay, got it? Is called gene. Will you keep in mind always? Tell me fast.
Yes, sir.
So Mendel's whatever we are having here. Okay, so Mendel's factor in modern term is called gene. So what he said, that the factor which is responsible for the tallness of the plant and the factor which is responsible for the dwarfness of the plant, okay, in F1 generation, they are not intermixed. They are not intermixed. Okay, why? Why? Because we are not getting any plant of average height. Like 6 plus 1, you will get 7, divided by 2, you will get 3.5. So there is no intermediate plant in between. We get all tall plants. Okay, got it? Got it, Jopita? Everybody in the class, got it? Okay, should I move forward?
Yes, sir.
And next was that when he did the self-pollination for the self-pollination. In self-pollination, we're going to make the only single plant here because self-pollination is done by transformation of gametes or pollen grains with the same flower. Okay, so this is self-pollination is being done. So this is the F1 generation. Okay, and when he did the self-pollination, doing this self-pollination, okay. So during self-pollination, he got the different results. So keep in mind always that when we are getting different results, both in F1 generation, sorry, both in monohybrid cross or dihybrid cross, if we are getting different result means self-pollination is done. Got it? So you have to keep in because there are several tricky questions usually come from this in the examination. So you need to keep in mind. Now, if I'm talking about this one, that this is what he obtained when he self-pollinated this one. So he obtained out of every four plants, okay, here he was getting three plants were tall and one plant was dwarf. Got it? He find three plants were tall and one plant was dwarf. Are you getting this point? This is three plants tall and one plant was dwarf. Got it?
Yes, sir.
So actually, if I talk about this one is that during the self-pollination, when he did the self-pollination, for every three plants, every four plants, three were tall and one was dwarf. This dwarf plant in the F2 generation, this is all F2 generation. This is all F2 generation. What does this indicate? What does this indicate? This indicate that when we are getting the dwarf plant, this indicate that there is a factor or gene that never forget its identity. Okay, we're going to discuss in this when we're going to do this in the gene level. Okay, gene level. So here is the conclusion of F2 generation. What he was having that for every four plants, three were tall and one was dwarf. When F1 generation was self-pollinated, then he got three tall and one dwarf plant. Okay, got it? Tell me fast. Got it?
Yes, sir.
Now moving to the next part here is how he explains. So let at genome level or at genotypic level, we are just taking that how he was explaining this one and how he was getting all tall plant in F1 generation and why he is getting three tall and one dwarf in the F2 generation. So let he explain how he explained him at this one. So he took the homozygous tall. So this is the plant which is homozygous tall, whose mother and father or whose male or female plants both are tall. And here this is homozygous dwarf. He take it a small t. Okay. And when they are separated, their gametes are formed, that this will be separated. This is the gene. This is one gene. Okay, or gametes. So this is he named it as homo. Homo means same zygous. Homozygous dwarf. This is homozygous dwarf. And he named this as homo means same. And this is homozygous dwarf. Sorry, tall. Okay. So this is now they are separated. Now when this capital T gene or pollen grain or whatever it is, when it fuses with this one, okay, we are going to have capital T and small t as a result. When this is going to have mixed with this one, we are going to have capital T, small t. Again, when this is going to mix with this one, we're going to have capital T, small t. And when this gene is going to mix with this one, this is capital T and small t. Got it? Got it?
Yes, sir.
Now, what he's saying here is that this capital T, small t, these are the two genes, two different genes. One is of tallness. This is of tallness gene, and this is of dwarfness gene. Okay. But But he see that still this plant is tall. Still, it has both gene. Okay, heterozygous condition. He called it heterozygous. Why? Because they are different. So this is heterozygous. Hetero means different. Heterozygous tall. He called it heterozygous tall. Hetero means different. Hetero means different. And still the plant is tall. So here the concept of dominant and recessive arises. The concept of dominancy and recessive gene arises. That this plant which is heterozygous tall, heterozygous tall. Okay. He named it, means it is going to have both the gene of tallness and dwarfness, but still the plant is tall. It means that the gene of tallness, gene which Mendel called factor of tallness is dominant, okay, dominant over recessive, Got it? Understood?
Yes.
That is it. This is a heterozygous condition. One is tall, there is a gene of tallness and a dwarfness, but still we will say that it is in a heterozygous condition, this plant is tall. So it means this gene is dominant, and this gene is recessive because it is not able to express itself. Got it?
Yes, sir.
So in F1 generation, we get all this type of plant, heterozygous tall. There was no homozygous tall. Okay. So this is this. And why they are all tall? Still they are going to have capital T and small t, both the gene of dwarfness and tallness. We are not getting any intermediate and we are getting tall. So we can see here that this gene capital T is dominant over the small t. Okay. Gene of dwarfness. And they are not mixing. He said that they are not intermixing. Why? Because you cannot see that if the capital T and small t mix together, they can form a gene like this. But they don't form the gene like this. They don't mix the gene like this. They don't form. The genes are not mixed up. They are not mixed up. They maintain their identity. How can we explain this one? That the gene are not mixed up because there is no intermediate plant is seen now. No 3.5 feet plant is observed. All plants were tall of approximately 6 feet. So this concept arises that the gene which is able to express itself is called dominant gene, and which is not able to express itself is called recessive. And they are not mixed up. They are not mixing. Why? Because we are not getting any intermediate plants. Are you getting my point?
Yes, sir.
We are not getting any intermediate. We are not getting any 3.5 pin plant. That is average of 6 plus 1. Okay, so this means that genes are not mixed up. Okay, and they maintain their identity. How we will prove this one? So let's took the F2 generation. So when he self-pollinate this heterozygous tall plant. Okay, so when he self-pollinate this one, so what he get? When he self-pollinate this plant, so he get capital T. Okay, this is capital T, homozygous tall. This is capital T, small t, then capital T, small t, and then small t. Got it? So since it is going to have both the gene of tallness, so it will be tall. Okay, so this will be tall, and we will got it homozygous tall, but it is tall. Okay, these two, since one gene is tallness is dominant over dwarfness, so these are also tall. Okay, but this is dwarf. There is no plant or no gene of tallness, so that's why this plant is tall. So actually in a heterozygous condition, you can see that this small t is not able to express itself because capital T is dominant. But it has not forgotten its identity of dwarfness, that this gene is responsible for dwarfness, but it is not forgetting it. Whenever the condition is satisfied for small t, we will simply say that that the plant will be dwarf. Okay, so it has not forgotten its identity. Are you getting my point? Are you getting my point? That it has not forgotten its identity. The dwarfness, gene of dwarfness has not forgotten. It does not mean that if it start living with this one, it has forgotten its identity of dwarfness. No. Actually, it is suppressed here. It is not able to express because this gene is dominant over the small t, over the gene of dwarfness. But whenever small t comes, the plant is dwarf. It means it has not forgotten. Understood? Understood?
Yes, sir.
Sure? Everyone? Understood?
Yes, sir.
Okay. Now, when he did the monohybrid and dihybrid cross, monohybrid cross, then he reached a conclusion and he gave a law that is called principle of dominance. What is called? Principle of dominance. And what does the principle of dominance state? Principle of dominance state that in a heterozygous condition, when there is an heterozygous, what is heterozygous condition? That when capital T and small t is there. This is heterozygous, means different. Hetero means different. This is homozygous, capital T or small t small t. This is homozygous. So he's talking about heterozygous condition. What he said, in a heterozygous condition, the two different gene or factor or trait remain together in an individual. They remain together in an individual and they are not mixed up because there were no average plants. They are not mixed up. Okay, like this. They are not mixed up. And he further added that the gene which is able to express itself is called dominant gene, and the gene which is not able to express itself is called recessive gene. You understood? Understood? And a question comes in examination here is that if I talk about the phenotypic means visible. Phenotypic means visible ratio. So phenotypic ratio of F2 generation is phenotypic ratio of F2 generation is 3:1 because three plants were tall, they were appearing tall, and one was dwarf. But if I talk about the genotypic ratio of Mendel's monohybrid cross of F2 generation in monohybrid cross, in monohybrid cross. So we have what? We have the ratio that is 2 is 1 is to 2 is to 2. 2 is to 1, sorry. 1 is to 2 is to 1. 1 is this one, 1, and these two are same. So that's why 2 and then. So this is the genotypic, means at general level, we can see the gene capital T, then small t, small t, small t, small t, and then small t. Sorry, capital T, small t, then small t. So this is the ratio of 1 is to 2 is to 1. Okay, this is the genotypic ratio and the phenotypic ratio is 3 is to 1. Got it? Cleared? Cleared?
Yes, sir.
Are you getting this one?
Yes, sir.
Mm.
Have you noted this one? This all? Have you drawn this one all?
Yes, sir.
This one drawn?
No, sir.
Draw it fast. Draw it. Done this one?
Yes, sir.
Noted? Okay, note this one also. F2 generation का था? Yeah, ये वाला draw कीजिए. Draw this one.
Done, sir.
What was principle of dominance? So note it down, please, everybody. Just a minute. So please note the principle of dominance here. Okay, please note it, a principle of dominance. Done this one? Principle of dominance, Jovita, Amma,
The writing.
writing, note it fast. The principle of dominance state that in heterozygous condition, when there is an heterozygous condition, the two different traits or factor remain together in an individual and they are not mixed up. Okay, why they are not mixed up? Tell me. Why they are not mixed
Because
up?
they're two different genes.
Because because we don't
One is
see.
because
We
its
always
physical
we
appearance.
see yeah, we see all tall plants. Okay, no average plants were there. That's why is say here that okay, the one which is able to express itself is called dominant trait, and the one which is not able to express itself is called recessive traits. Note it fast.
Done, sir.
Okay, everyone done?
No, sir.
Now the second law Now the second law, what he said actually for this monohybrid cross, he gives another law, that is law of segregation. Law of segregation or law of separation. Law of separation or law of purity of gene. We called it law of purity. Law of purity of gene. And what does it state? It state that in a heterozygous condition, again, in a heterozygous condition, okay, the two different traits remain together in individual. The two different gene remain together in an individual and they are not mixed up. The same thing from the law of principle of dominance, the statement is same. In a heterozygous condition, the two different traits remain together in an individual and they are not mixed up. Now here the change, they maintain their identity. How we can say that they maintain their identity? That in F2 generation, when we have capital T, capital T, small t, capital T, small t, capital T, small t, and small t, small t. So this is tall, but this is dwarf. So they are not forgetting their identity. Okay. Whenever the condition is satisfied with the small t, the dwarf plant is there. So it means, it does not mean that if the gene start living with capital T and small t, it will forget its identity of dwarfness. No. Whenever the condition is satisfied, we will have a dwarf plant. So what does the law of purity of gene states? That in a heterozygous condition, the two different traits remain together in an individual. The two different traits remain together in an individual and they are not mixed up. They are not showing. And how can we say they are not mixed up? Again, because there were no average plant. If they mixed up, they will give an average plant, but there is no average plant is there. So they are not mixed up. They maintain their identity. Are you getting my point? They maintain their identity. Okay. And during the time of gamete formation, when the gamete formation is taking place, okay, then only one gene receive a particular type of gene. Sorry, one gamete receive only a particular type of gene. For example, like in human beings also, we have 46 chromosomes. Out of this 46 chromosome in human beings, we have 44 autosomes, and one X and one Y. These are called the sex chromosomes. Okay. But when the gamete formation is taking place in human males, okay, gamete formation, sperm is going to have, then during that condition, we will going to have 22 autosome or either X or either Y. A chromosome, a sperm or a gamete cannot have both X and Y. Half number of chromosomes will be there. Are you getting my point?
Yes,
Are
sir.
you getting my point? That during the time of gamete formation, for example, if this is a heterozygous condition, so when the gamete formation of this is taking place, then each gamete will receive either this or either this. There is no gamete which is going to have both the genes. Got it? Got it? So what does law of segregation states? The law of segregation states that in a heterozygous condition, the two different traits remain together in individual and they are not mixed up and they maintain their identity. Mixed up? Why not? Why they are not mixed up? There is no average plant. And they maintain their identity because when small t is there, then a dwarfness is there. So it means it knows its identities. The gene of dwarfness didn't forget its identity. It still has the ability to dwarf the plant. So it has maintained its identity. And when the gamete formations take place, then each gamete receive a particular type of gene. Okay, got it? Got it? Got it?
Yes, sir.
Yes, sir.
So let you write what is law of segregation. Just a minute. You write please law of segregation.
Done, sir.
Done?
Yes.
Everyone? Done? Should I move forward?
Yes, sir.
Yes, sir.
Okay, just have some questions on this topic. Okay. Here we have a question. If the question comes in examination, that how do Mendel's Mendel cross or experiment shows that traits or trait can be dominant or recessive. So how does it shows trait can be dominant and recessive? So when he crossbreed a homozygous tall plant and a homozygous dwarf plant, they were all tall plants. Okay, they were all tall plants and there were no intermediate. Okay, this indicate that that a gene is dominant, means one gene is dominant over the other. Okay, got it. And how his experiments show that the traits are not intermixed. Sorry, how the traits maintain their identity, how they are they maintain their identity. So in F2 generation, he was having capital T, then capital T small t, then capital T small t, and then small t. So here we are getting a dwarf plant, and this dwarf plant shows that traits are not intermixed, they maintain their identity. This dwarf plant is showing. Okay, so if the question comes in examination that how do Mendel experiment shows that traits may be dominant or recessive, so you can explain it with the monohybrid cross, that Mendel crossbreed a homozygous tall plant with a homozygous dwarf plant. He got all tall plants. Okay, there was no average plant and no intermediate or no dwarf plant was there. So it means that the tallness gene is dominant over the dwarfness. Okay, and how do Mendel experiments show that traits are not traits maintain their identity? So you have to explain it with this one, that in F2 generation of monohybrid cross, Mendel find 3 talls and 1 dwarf plant. Okay, so it means that this dwarf plant indicate that that genes are not intermixed and they maintain their identity. Okay, got it? Got it? Got it? So please
Yes,
note
sir.
it down. Note it down. Just a minute. Note it down. So note it down here. How do Mendel experiments show that traits may be dominant or recessive? So in monohybrid cross experiment, there were tall plants in F1 generation, no intermediate. But in F2 generation, for every 4 plants, 3 plants were tall and 1 was dwarf. This show that genes may be dominant or recessive, as there were no mixed up and they maintained their identity. Don't write it in bracket. Just remove this one here, bracket, and write it as dwarf plant appear in F2 generation.